Hello, everybody, and welcome to Thursday, September 17th, 2020. It's a great day to be a junior Potter here. It's the last day of the week as far as regular science class goes. Tomorrow is Leadership Academy, so for your agenda today, it's the bell ringer. Make sure you've done all of them for this week. That way, when I go in there, I can get your credit for every single day for participating through those bell ringers there. And without further ado, we're going to go through day 2 of the microscope tutorial. And I've been busy plugging away with our microscope. You can kind of see it here in the corner. Let's see if I can move the... Yeah, I've got a microscope hooked up to the computer here, so maybe we can do a little bit of microscope action so you can see some of that stuff here. I've been sorting through all the slides for next week to make sure everything's been back and put together so that we can actually, enjoy ourselves next week and know where everything is at. So without further ado, what you can do at home for our microscope tutorial, we'll get that brought up here today. So it's the same assignment as yesterday. So go to the Wednesday, Thursday folder in Schoology, look up the assignment microscope tutorial, and then when you have it opened up, this is what you should be seeing when everything's all said and done with it. You're going to do 5 different activities, and for you at home, you might not be able to do as many of these. I think there's 3 of them that you'll be able to do for certain. Some of the other ones, unfortunately, you're not going to be able to do too much with. We'll run through everything that we have here. On slide 13 here, you're going to be talking about magnification and resolution, so you need to read through that following thing and then answer the question that my face is currently covering over right now. But it's right below that. Let's see if I can change it a little bit there. So see whenever there's that green section, make sure that you're doing those areas there. This one's talking about magnification, so I'll see if I can get it loaded up here with the webcam. Right there, you'll see that on the eyepieces in any type of microscope, they'll have a number with an X at the end of it. That's how much stronger it is to your normal vision. So this one right here says 10X. That means it is 10 times the magnification, or how close you see something, than normal. Now what you could do, you could just theoretically put the eyepiece up to you and kind of use it as a magnifying glass, but that just zooms everything in. It does not really do the clearing up of the image, which is what our microscopes handle. That's called resolution, so we'll have to add a little bit of resolution to it to make it clear and not as blurry of an image. What you'll be doing next, you won't be able to do this at home, but I'm going to show you anyway. So I'm going to detach one of our lenses here off of the microscope, but on each one of the 3 objective lenses, low, medium, and high, you'll have the same kind of numbering system there. So you can kind of see that there's a 4 happening there. That means this lens is 4 times the normal power. You'll also see that each one of them is color-coded. This one's kind of got a red ring around it. So in this case, if you were here, you'd be able to grab one of our microscopes and decode that this was a red, representing low power. What you'd be doing next is you'd be calculating the total magnification. So when you look at our microscopes, I'll pull this one back off again. You've got not only your objective lens that you're taking a look at, but you're also taking a look at the eyepiece because it's its own lens itself. And you can see that it kind of makes a little math problem. 10x4, 10 times 4, you calculate that out. That means when using the microscope on low power, you are getting 40 times your normal eyesight because you multiply the number that is on the lens by the number that is on the eyepiece, which is pretty nifty. That way you can see 40 times the normal eyesight there. Usually medium power is 10, so usually we get 100 times normal because the eyepiece is always 10, so 10 times 10 is 100. High power, usually with some of our microscopes, they range from either 40 times to 60 times. So that's somewhere between 400 times your normal vision or 600 times your normal vision if you're using some of our other microscopes. Some of them have a little bit different power there with it. Adjusting the focus. You're going to adjust the focus using the coarse adjustment knob. And I'm going to try something right here just to see how it works. I'm going to bring up the microscope itself, because I've got it all attached up here to work with, and we're going to see if we can zoom in on something here and get it actually able to show up. So when you first put something underneath the microscope, it may look like this. It is a blurry mess. You cannot see anything whatsoever with it. What you're going to do, you're going to adjust the coarse adjustment knob, which is the big knob, and it is going to bring it closer or further away. As soon as you see that it's brought in focus, you need to stop, and boom, there we go. We're actually looking at pollen right now off of a plant. And if it's still not all the way clear, if they have a fine focus, you can twist the fine focus and get it adjusted more. Now this is on low power. You can then switch it to medium power. Carefully adjust it bit by bit until you get those kind of in play there. So there's a closer look of pollen. You'll notice that they're all red. That's just the stain that they use with it. This is underneath high power, so you can really start to see some of the cellular work with the pollen grains. They're happening with it. You can kind of see the nucleus is that little middle dot there of the cell. You kind of have the cell membrane going all the way around them. These ones are actually composed of many cells, so there'd be multiple cell membranes happening there. But you can kind of see a little bit of what it has. It depended on if they got sliced open or not, as far as what we can see, because some of them just show the outside of the pollen, which is what this one in the middle is kind of showing, the outside of the pollen grain. You can't really see the nucleus or anything in that one because that one didn't get cut open, sliced down the middle. You can kind of see different aspects of it though as I tweak it back and forth. You can see how pollen is kind of bumpy. That's so it can attach to the bees. So you can kind of bring the fine focus back and forth to see different aspects of whatever you're looking at with it. But that would be how you would adjust things using the adjustment fine focus knob there. And then transition back over. The light sources, we use two different light sources. Some of them use a mirror, some of them use the actual physical light source itself. So 2 questions just asking about how you would be able to figure out which one's which on there. Adjusting the light if we need less or more light. And then looking at your first specimen, that'll be something we'll have to take care of in class since you don't have a microscope. If you want to draw the pollen grain that I just did there, you're more than welcome to do so. That's just one of them there, so I'll put it back up there. If you want to draw that as one of your drawings and take a picture of it. But usually a lot of the time, that's what we end up doing is drawing what we see. Let me I'm gonna put back up. Here's some salt as far as what salt looks like. You can see that there's little cubes of salt. Try and take a 2D image, make it 3D there. Just cubes on top of cubes. It's all salt crystals. It's the stuff that, next to pepper there, salt and pepper. Little tiny cubes, little salt crystals magnified up. If you look under it under low power, you can kind of see some of the crystals happening there. Might be hard to focus since I'm on camera and trying to focus at the same time. I'm not actually looking through it. I'm looking at just the image that's popping up there for it. So all those little tiny salt crystals everywhere, zoomed in enough, we end up getting more of what they actually look like, those little cubes happening with it. This is more showing just the microscope ends of it. Some scratch marks there, some dust obviously is on there because I just grabbed one of these that we were working with. Yeah, that's just more dusty than anything. So you get the idea and the point. You're working through our microscope tutorial stuff today. Hope you're staying safe out there. Tomorrow's Friday, Leadership Academy. Maybe I'll see you back here in action for Leadership Academy, but stay safe out there. Hopefully we'll see you again real soon there. If you have any questions whatsoever, always remember you can email me, but stay safe, take care. Email me if you got questions, Try to work on as much microscope stuff that you can. Since you don't actually have a microscope, there are some things that you can read and do. Bye!